inviting me to speak here today and tell you a bit about some of the research that's going on in the structural biophysics group at Cardiff University. So today I'm going to be talking about the structural basis of keratoconus and how corneal cross-linking can help to stop keratoconus progression. So the cornea, it's responsible for about 70% of the focusing power of the eye. And to function properly, it needs to be transparent to allow the passage of light through the eye so it can get through to the retina. It needs to be precisely curved to focus the light onto the retina. And it needs to be strong to resist impact and maintain the curvature of the cornea. And the cornea is unique in that it's the only tissue in the human body that has to combine strength with almost 100% transparency. So because it's transparent, the cornea might appear to lack substance, but it's actually a highly organized group of cells and proteins. And if we have a look at the structure of the cornea here, we can see that it's comprised of 5 very distinct layers, which from the front to the back, we've got the epithelium, then we've got Bowman's membrane, the stroma, Descemet's membrane, and the endothelium. And each of these layers plays a really important role in the functioning of the cornea. So, for example, as Martin mentioned earlier, the epithelium, it provides a protective barrier against infection and injury. And the endothelium that's at the back of the cornea, that helps to regulate the water content of the cornea. So it stops it from swelling, because if it was to swell, then it become cloudy and wouldn't allow light to pass through it. And then in the middle, we've got this big layer called the stroma. And this layer, it's about 90% of the thickness of the cornea. And this is the layer that provides the cornea with most of its strength. And it's also the layer that all of our research is done on. So changes in the structure of the cornea can lead to changes in the functional properties of the cornea. So it can affect the strength, the shape, or the transparency of the tissue. And all of these changes can lead then to a reduced quality of vision. Now in keratoconus, the mechanism by which the tissue becomes progressively thinner and weaker and gradually becomes misshapen, it's not fully understood, but as we've heard in the talks earlier, it's likely a combination of factors. So there'll be genetic factors, environmental factors, but there's also changes in the structure of the cornea, which are involved in the progression of keratoconus. So, like all tissues in the human body, so from skin and tendon, cartilage, even bone, they all owe their strength to a protein called collagen. And the cornea is no different in this respect, except in the cornea, there's a very precise arrangement of collagen. And this allows the tissue to be both strong and transparent. So in the corneal stroma, you've got very strong collagen fibres that are arranged in layers, in very thin sheets called lamellae. And there are about 250 of these collagen layers, or lamellae, throughout the thickness of the cornea. And within each of these layers, the collagen fibrils, they're regularly spaced, and they lie parallel to each other. But they do change direction throughout the thickness of the cornea. So hopefully you can see this in the microscopy image on the top right there. We've got 5 collagen lamellae. And in some of them, so up near the top, you can see that the collagen fibres, they've been cut in cross-section. So they appear as little blobs, but they would effectively be coming out of the screen towards you. And then in the next layer, they've been cut longitudinally. They're effectively going left to right across the screen. Now, this kind of layered arrangement, you can think of it like the structure of plywood, in that you've got lots of thin sheets of wood that are all arranged with the wood grains running at different angles to each other. And the strength of the plywood is dependent on the strength of the individual layers, the number of layers, and also the direction of the wood grains relative to each other. And it's very similar in the cornea. This layered arrangement of collagen helps provide strength to the cornea. So the arrangement of these collagen layers, it's not uniform throughout the entire thickness of the cornea. So In the middle and back of the cornea, we get this plywood-like arrangement that I showed on the previous slide. But if you look at the microscopy image up at the top there, you can see that as you move towards the front of the cornea, the lamellae get more and more interwoven. And this interweaving, it helps to provide strength and stability to the structure of the cornea to help it maintain its shape, in a similar way to how birds kind of interweave fibres to provide strength and shape to their nests. And then at the bottom here, we've got two more microscopy images that were taken from the front of a front stroma of a healthy cornea and a keratoconus one. And you can see that in some places, the collagen lamellae, they're not always continuous along their length, so they split and they change direction. And this kind of point where they split, it's potentially a weak point. And in keratoconus, you've got a lot more enzymes chunking away at the collagen. So you can imagine it be a point at which tearing might occur. And in keratoconus, we get a lot less interweaving of collagen, so it's a lot more readily tearable, I guess. And you can think of this like a tight pair of jeans, that if the stitching's strong and the material's strong, then they'll hold together. But if the stitching's weak and the material's weak, then they can start to split and then they just keep on going. And in the cornea, the forces that are acting upon it are intraocular pressure. You've also got the action of the eye muscles during eye movement. And in case of keratoconus, you've also got eye rubbing. And the normal cornea would be able to resist these forces, but of course the structure of the keratoconus cornea is that much weaker and it more readily yields then to shape changes. So in the frontmost part of the cornea, or in the corneal stroma, you've got some collagen lamellae that insert into Bowman's membrane. And these insertion points, they're kind of anchoring points, and you can think of them like tent pegs, that they help to hold the structure in place and maintain its shape. So like with a tent, if you've got tent pegs in, when the wind blows, it still maintains its shape. But unfortunately in keratoconus, there's fewer than normal of these anchoring points. And this means that it's more easy for the collagen lamellae to kind of move around and alter their position. So now if we look even deeper into the cornea at the individual collagen fibrils, we can see that, this schematic is quite complicated, but the only thing that you really need to notice is that each of the levels, going from molecule right up to the fibril, there's an alternating twist at every level. And that's similar to the structure that you see in rope. And this alternating twisted structure, it provides the collagen fibrils with more strength. And in the cornea, the collagen fibrils, they've also got chemical bonds within them. They kind of act like a glue, helping to hold the structure together and provide it with more stability. But in keratoconus, we've got fewer than normal collagen bonds, so it's effectively less glue holding the structure of the collagen fibril together. So there are many ways in which we can study the structure of the cornea, and the choice of technique, it really depends on the size of the structure that you're wanting to look at. So, for example, you've got light microscopy shown up on the left there, and that provides really nice images showing the whole tissue thickness. So you can see the epithelium at the top all the way down to the endothelium at the bottom. Then we've got electron microscopy, and that gives a very localized view of specific regions of the tissue, so you can focus in on cells within the cornea, or the third image along there, you can see the collagen fibrils within the corneal stroma. And then there's X-ray scattering, which provides us with numerical information about different collagen parameters, so things like the size of the collagen fibrils and their orientation. So the two main techniques that we tend to use in the structural biophysics group at Cardiff University are electron microscopy and X-ray scattering. Now, electron microscopy, it provides a very localized view of the tissue, as I mentioned previously, whereas X-ray scattering, it provides a measurement throughout the whole thickness of the tissue, so you get an average measurement of all the different parameters. And we collect our X-ray scattering data at Diamond Light Source, which is the UK synchrotron radiation facility in Oxford. And the way that we collect the data is that we take a human donor cornea, position it into a sample holder, and then direct an intense beam of X-rays through it. And then we look at the way that the X-rays are scattered as they come out of the other side. And by looking at the X-ray scatter pattern, we can get really useful information about the structure of the cornea. So we can find out the average spacing of the collagen fibrils. We can find out their diameter, and also the amount and direction of collagen at a particular point in the tissue. And all of these structural parameters all tie in very closely with the functional properties of the cornea, so its transparency and its strength. So by collecting X-ray scattering data at very fine intervals across the cornea, we can produce these maps, shown here in the middle, which shows the predominant direction of collagen across the human cornea. And the thing to bear in mind with these maps is that we're looking at just the predominant direction of collagen. So it's over and above a background where collagen is going in all directions within the plane of the tissue. So in the center of the cornea, you'll see there are these cross-shaped plots, and that tells us that there's more collagen lying in the vertical and horizontal directions than in any other. And collagen, it's strongest along its long axes. So if there's any preferred orientation of collagen, then this is going to provide increased strength in that direction. So on the image on the right here, I was just showing that it's interesting to see then that these preferred directions, they line up with the insertion points of the major eye muscles. So it's possibly providing increased strength in these directions to resist shape change during eye movement. And just the other thing that I want to say from this map showing the direction of collagen is that the plots in the center of the cornea, they're all fairly uniform in size. And that tells us that the amount of collagen in that region is fairly uniform. So if we look at the situation in keratoconus, at the top of the slide here, we've got a map showing the predominant directions of collagen in the healthy cornea that we saw from the previous slide. So we've got mostly collagen traveling in the horizontal and vertical directions. And then at the bottom there, we've got two keratoconus corneas. And these were from people that were undergoing corneal transplant surgery for keratoconus. And we were provided with the tissue that was removed. So it's just the central kind of 7 to 8 millimeters of the cornea. So we never actually know what goes on beyond that region. And also the other thing to note is that we're also getting tissue that's at a very advanced stage by the time we get to look at it. But what you can see from the plots at the bottom is that in some places there's very, very tiny plots or non-existent plots. And this is telling us that there's not very much collagen at those positions, which is consistent with the fact that you get a lot of thinning at the apex of the cone. But the direction of the collagen is also altered. And this is consistent with the idea that you've got collagen layers are changing direction, they're slipping and they're sliding away from the apex of the cone. So here we went on to look at another 5 keratoconus corneas in quite a lot more detail. Sorry, it's probably too small for you to see some of this. But on the left here, we've got topography maps showing the surface elevation of 5 keratoconus corneas. So we've seen from the previous talks that the region of greatest elevation in these ones is the kind of purpley reddy region, and that's been outlined on the topography maps. And then next to it, we've then got topography maps showing the thickness of the cornea. So the thinned region is shown in the pinky red colour. And then in the middle, we've got maps showing the predominant orientation of the collagen, and next to it then maps showing the distribution of collagen mass. And as with all keratoconus studies, there's a lot of variation between specimens. But the main thing to note was that the most severe case, which is the one shown on the bottom row, that showed the most significant changes in collagen orientation, and there was also a lot of changes in the distribution of the collagen mass in that corneal button, whereas in the least severe case, in terms of corneal thinning shown at the top, there were very minimal changes in the orientation and distribution of the collagen mass. So when you put all this data together, it really convinced us that that keratoconus progression, it does involve both a slippage and a redistribution of the collagen lamellae. So our collaborators in the University of Birmingham, Alabama, they went on to use our X-ray scattering data to perform numerical modelling to try and predict the most likely mechanism of keratoconus progression. And what we have here is on the left, we've got topography maps for a healthy cornea. So it's got a normal corneal thickness and normal surface elevation. And on the far right, then, we've got similar maps for the keratoconus cornea, and that's the most severe case of keratoconus from the previous slide. So in the first model, they kept the thickness of the cornea at a normal level, and they just incorporated the abnormal collagen orientations that we detected from the X-ray scattering data. And if you look at the surface elevation map at the bottom for model 1, you'll see that it doesn't match the real-life keratoconus data on the bottom right. So in the second model, they went one step further. They included the abnormal collagen directions from the X-ray scattering data, and they also reduced the thickness of the cornea to match that of the keratoconus data. And again, still the surface elevation map didn't match the real-life data. So in the third model, they went one step further. They included the actual thickness of the keratoconus cornea, the abnormal collagen directions, but this time they also included in the model the ability for the lamellae to slip and slide. And by adjusting the amount by which they could slip, they were able to produce a surface elevation map, which I think very closely matches the real-life data. So this modelling, it adds further weight to the belief that keratoconus progression involves both a loss of collagen and also a redistribution of collagen mass within the tissue. So I mentioned earlier that most of the protein in the corneal stroma is collagen, but at the very back of the corneal stroma, there's a region that's has very different biomechanical properties to the rest of the cornea, and this region contains elastic fibres. So elastic fibres, they're present in lots of tissues, and so, for example, they're present in the lungs, and they act like springs, so that it allows the tissue to stretch, but then to quickly recoil back to its original shape. And when we look at them with transmission electron microscopy at the back of the corneal stroma, we can see all the little tiny black dots there, the collagen fibrils, and then these big black blobs are the elastic fibres. So at Cardiff University, we're very lucky to get a fancy bit of kit called a serial block face scanning electron microscope. It's a bit of a mouthful. But it allows us to, well, for the first time really, to be able to look at these elastic fibres in 3 dimensions throughout the thickness of the cornea. And sorry, Andy, could you please play the video? I'll just show you how it works. So it images the surface of the cornea, and then it will take a very thin slice and then re-image the next surface. So in this way, then you can get thousands of images throughout the thickness of the cornea, and they can then be put together to build a 3D reconstruction of the tissue at the particular things that you're wanting to look at. Okay, so here we've got another video reconstruction of the back of the corneal stroma, and this one's taken towards the white of the eye, so just the boundary between the cornea and the white of the eye. And could you play the video, please? Thank you. Thanks. So at the back here, we've got the blue part is Decimase membrane, and we've got the elastic fibres shown in yellow, and we've got a corneal cell shown in pink. And you can see that there's all the elastic fibres, they're all concentrated towards the very back of the corneal stroma. And these elastic fibres, here, I'll just say, as we go we're now moving towards the centre of the cornea, and you can see that the elastic fibres, they start to split and gradually get thinner as they move in towards the centre. So at the edge of the cornea, these elastic fibres, they're what we call true elastic fibres, in that they contain a very stretchy elastin core surrounded by a more rigid outer layer. Okay, so you can do lots of pretty videos and images with this technique. I'll move on. So here we've got another video reconstruction showing the elastic fibres again in the very back part of the corneal stroma. But this time it's taken from the centre of the eye. And we can play the video again, please. Thank you. Oh, how's it going? Not sure it starts. Oh, there we go. So again, in the centre of the cornea, you can see that the elastic fibres shown in yellow, they're located right towards the back of the cornea. But in the central part, they're much thinner than we saw out in the edge of the cornea. And these ones in the centre of the cornea, they haven't got a stretchy elastin core to them. So they're much thinner and they're less stretchy than the ones that we saw at the edge. Okay. So our group went on to use all this information about the elastic fibres to generate a model showing the elastic fibre network in the healthy cornea. And here we can see at the edge of the cornea, you've got stretchy elastic sheets. And then as you move further in towards the centre, the fibres, they gradually start to get thinner, and they also start to lose their elastin content, so they get less stretchy. But what they do is that they form connections that go across the whole cornea, so at the very back of the cornea, there's a whole network of them. And this allows loads to then be transmitted across the cornea. But obviously, because it's more stretchy at the edge, you'd expect much more give in that region. So what's the situation in the keratoconus cornea? Well, we've seen already that there's major disruptions to the collagen organisation, but we also want to know what was happening with the elastic fibres and whether they were also altered in keratoconus. So again, we're very thankful to two more keratoconus patients who are undergoing corneal transplant surgery. They provided us with tissue for us to investigate. And here at the top, we have images showing the elastic fibre network in the healthy cornea, this is taken from the centre of the cornea. And then at the bottom, we've got the keratoconus cornea, also from the central region. And you can see that in the healthy cornea, we've got a huge expanse of elastic fibre networks, or elastic fibres, all kind of concentrated towards the back of the cornea. Whereas in keratoconus, there's far fewer elastic fibres, and they're also much thinner than the ones that we saw in the healthy cornea. So if we now go back to this model of the elastic fibre network in keratoconus, and if we take out some of the elastic fibres from the centre to represent the situation in keratoconus, then it's possible to imagine that you've got this fewer connections across the cornea with the elastic fibres. You've also got an altered collagen orientation and disruption to the collagen. So you can imagine that this will affect the biomechanical properties of it and make the tissue weaker. But as I said, we still don't know what's going on at the edge of the cornea. So we don't know if the collagen organisation is altered in that region, and also if the elastic fibre network's altered. But I think we can probably assume that there probably are changes that extend beyond the centre, and this, again, would further impact the biomechanical properties of the tissue. So just to summarise, in keratoconus corneas, we've seen that there are fewer collagen layers in the tissue. There's less interweaving of the collagen layers in the frontmost part of the cornea. and there's fewer collagen anchoring points to hold them in place. We've also seen that the organization of collagen is altered, so they change directions, and it's consistent with the idea that we've got layers sliding away from each other, and that there's an abnormal elastic network in the keratoconus cornea. So all of these things contribute to the keratoconus cornea being weaker and less stable than the healthy cornea. And that's why it's really important that the patients are urged to try and resist the urge to rub their eyes, because just the mechanical shearing effect that this would have on the already very weak structure. So in relation to that, then, there's a recent study by Cardiff Group in Switzerland, where they looked at the mechanical forces that were exerted on the eye during eye rubbing. And they found that with knuckle rubbing, the force exerted was about 2 to 4 times higher than with fingertip and fingernail rubbing. And the forces were quite high. So it's probably very hard to resist the urge to do it, but it's hopefully this will help. So the question is, how can you stop keratoconus progression? Well, we've seen in the previous talk about how since the introduction in the late 1990s of riboflavin UVA corneal cross-linking therapy, that it's been shown to be highly effective at stopping keratoconus progression. So the technique, just to recap, it involves removing the top layer of cells from the cornea, so the epithelium, and then applying riboflavin or vitamin B eye drops, which then are allowed to soak into the corneal stroma, and then exposing the cornea to UVA light, and in this way we form crosslinks. So we just look at how does it work. So does corneal cross-linking strengthen the corneal collagen? Well, if we go back to this schematic here, where we talked about how collagen bonds in the collagen fibril provide it with strength, well, we know that as the healthy cornea, as it ages, it gradually becomes stiffer with age, and this is as more crosslinks are formed within the collagen fibril. But with keratoconus, the tissue starts off with fewer collagen bonds to start with. But we've shown that by cross-linking the cornea using riboflavin and UVA, we can generate new crosslinks to form within the collagen fibrils and around them. And so then this has the effect then of strengthening the collagen, which is one way in which it can help to stop keratoconus progression. So does cross-linking prevent slippage of collagen layers? Well, scientists have shown that it doesn't actually make it any harder to separate individual collagen lamellae after cross-linking. So it doesn't improve the cohesion of the collagen lamellae. But if you look at the graph at the bottom, you can see that this is with strips of cornea, strips of human cornea. Some of them were cross-linked, other ones were left untreated. The cross-linked ones were 4.5 times stiffer than the untreated ones. So by increasing the overall stiffness of the tissue, that's one way in which keratoconus progression can be halted with riboflavin UVA cross-linking. So does cross-linking improve the resistance of the cornea to enzyme attack? Well, we know that in keratoconus there's a greater number of collagen-attacking enzymes that are present in the cornea. And what we've shown is that in our laboratory studies is that when you put an untreated cornea and a cross-linked cornea, if you put them into an enzyme digest solution, come back 12 days later, you find that the untreated one has completely disappeared and the one that had cross-linking is still relatively intact. So there's a very pronounced increase in the resistance of the cornea to enzyme attack after cross-linking. And this is another way in which it stops the progression of keratoconus. So it's the technique's working by stiffening the cornea, but also improving its resistance to the enzymes. So another question that's often asked is, how long does cross-linking last? Well, it's impossible to answer conclusively at this stage because there's still kind of waiting for more clinical trials to be done. But so far, the long-term clinical trials, the results look fantastic. So that you're seeing stability in most corneas up to 13 years after treatment, and there's nothing to say that it's not going to go on for many years after this. It's just that's as far as the clinical trials have gone at the moment. But from a structural point of view, I think it's important to say that the rate of collagen turnover in the stable cornea is incredibly slow, so it takes many decades. And with riboflavin UVA cross-linking, we're increasing the resistance of the cornea to enzyme digestion, so we're making the cornea more stable. So I think that that we'd also expect the keratoconus cornea that's been treated to last for a long time before it progresses. So just to show you an example of how slow the collagen turnover is in the cornea, here we've got a map on the left showing the predominant directions of collagen in a human cornea that underwent corneal transplant surgery 13 years previously. And you can see where the red arrow is there, that's highlighting the boundary between the host cornea and the donor cornea, which is still very obvious after 13 years. And we also have similar data showing the same effect after 28 years. And in the 28 years one, we can also see that the donor cornea was put in an abnormal orientation with respect to the rest of the cornea, and that still seems to be present after 28 years, so it's not been completely remodeled. So I think that there's a lot of hope for riboflavin UVA cross-linking being a very long-term treatment for keratoconus progression. So I hope I haven't raced through that too much, but I just want to acknowledge the Medical Research Council who funds our research, and also our research team. So we've got Dr. Sian Morgan, who's with us here in the audience, and speak to you later. She does a lot of the cross-linking work. And we've got Dr. Phil Lewis at the front there. He's our electron microscopist. So he did all the lovely reconstructions. And James Bell, who's our biomechanics expert, and Keith Meek and Andrew Quantock, who lead the corneal research program at Cardiff University. Also, we get a lot of support from Diamond Light Source, who helps with our X-ray scattering data collection, and also the clinical members of the UK Cross-Linking Consortium provide us with a lot of advice and support, and in particular, Professor David Bart and Professor Stephen Tuft, who've always been very supportive of our keratoconus research program at Cardiff. And also finally, thank you very much to all the keratoconus patients that have donated tissue over the years to our research to make it possible. So thank you.